Welcome to the lame fired lesson. In this lesson, we're first going to go through some of the questions on the do now that we didn't get to go over because the alarm. And then we're going to take a closer look again at the G1S checkpoint, review the G1S checkpoint a little bit, and then talk about the signal transduction pathway that focuses in the G1S checkpoint. So if we're looking at the questions on the do now that are here, I'm going to actually focus on 1, 2, 4, and 5 because question 3 we focused on in class when we were learning the lesson with the 15 minutes that we had. So we're going to go through the do now. So the first question on the do now is explain how bacteria use quorum sensing to launch a successful attack on their hosts instead of being killed by the host. Quorum sensing allows the bacteria to wait until there's enough of them to make a difference and be able to kill the host. If there wasn't enough of them, the host would simply defeat them, and they wouldn't be able to make the host sick. Now, how this works is that when there's a small amount of bacteria, they don't make enough signaling hormone, or inducer it's called, and there's not a lot of it around because they're not making as much of it, and it's unlikely for the bacteria to encounter the inducer molecule. But when there is a higher density of bacteria, or more bacteria secreting that molecule, it's more likely to bind to the receptor. And when it binds to the receptor, it activates genes that allow them to attack. But again, that molecule is only likely to bind to the receptor when there's a lot of it. And that's only likely to happen when a lot of bacteria are present producing it. So quorum sensing allows the bacteria to wait until there's enough of them to participate in successful group behavior. Now, if we look at the second question asks, how did the professor in the video say we could develop new antibiotics? Well, I have drawn a picture for you to look at. And in the picture, we have our bacterium here. So we have our bacterium here, 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 and here. And we see on their cell membrane they have a red receptor. And then the blue molecules are the quorum ligands, the ones that activate the pathway. What the professor suggested in the video is to design inhibitors of the receptors. So that instead of the quorum sensing molecule binding to the receptor, the inhibitor will bind to the receptor and block the presence of the quorum molecule. Now, why will this stop the bacteria from making you sick? Well, if you remember, the bacteria only launch the pathway when they bind to the receptor and activate the genes to launch the attacking pathway. So if we're able to build these green molecules, in this case, that can block the receptor, the bacteria won't be able to talk to each other, and essentially they won't be able to count each other and say, okay, there's enough of us to launch our attack. So these receptor molecules, or these inhibitor molecules, will block the quorum-sensing molecules from getting into the receptor and prevent the bacteria from sensing that there's enough of them to attack. And they won't be able to launch the protocol, or they won't be able to make the genes that will allow the bacteria to attack and therefore make you sick. The last two questions on the do now are easier to answer and quicker. So the first question is, what two things must cells do to meet the goal of mitosis? If you remember, the goal of mitosis is to make identical cells. And the two things that cells must do to meet the goal of mitosis is they must replicate the DNA, and then they also must divide it evenly amongst the cells. So mitosis meets those two goals of replicating the DNA and then dividing it equally amongst the cells. Question 5. Explain how microtubules help the cell progress from prophase to anaphase. So microtubules, which is the same as saying, or which make up spindle fibers, are made in prophase. And once they are fully made in prophase, the spindle fibers, or microtubules, attach to the chromosomes at the kinetochore, and they elongate to move the chromosomes to the middle. So that's what they do in prophase, is they're made in prophase, they attach to the kinetochore of the chromosome, and then move the chromosomes to the middle. That is now metaphase, okay? To enter anaphase, the microtubules begin to shorten, which is called depolymerization, and the shortening of them allows you to pull the chromosomes to opposite ends of the pole, which is necessary in anaphase. So hopefully, if you're doing this correctly, you have looked at your do now, which you hopefully were able to complete in class, and added to your do now. I am now going to upload this video, and start a new video for the G1S checkpoint, because I don't want to run out of time. These videos can only be 15 minutes long on the free software. So I'm going to upload the video, and you'll get 2 links, or you click on the second link when you get it.